pleased to introduce chromatid and to review how to gather the most complete picture possible of genomic changes in your samples. We'll look at several well-established analysis options, then we'll place chromatid's DGH assays within that context. 3 different lenses will help put this into focus: analysis of a single very abnormal cell, analysis of a mosaic sample, and analysis of a sample set. We'll then close out by looking at how orthogonal data sets can plug gaps through which critical pieces of information may otherwise slip by. Chromatid is based in Longmont, Colorado, just outside of Boulder. We are a research-focused team committed to providing top-tier tools, service, and expertise to advance scientific progress. We work with scientists in academic institutions, in large pharmaceutical companies, and within governmental settings. Chromatid supports the scientific community with a wide range of genomics tools and methods. These include custom probes, genomic integrity G-banding, and a sophisticated set of proprietary assays, which we'll look at shortly. Let's now take a high-level look at some of the most widely used cytogenetics methods. Array-based comparative genetic hybridization, ACGH or array CGH, places DNA from lysed cells on a glass slide, which has DNA probes immobilized onto it in a grid pattern. The sample DNA and probe DNA are each labeled in a different color, which enables estimation of copy number differences based on the ratio of fluorescence wavelengths. G-banding relies on staining differences between chromosomal regions, which are relatively AT-rich or GC-rich, looking at one cell at a time. The signature banding patterns allow identification of chromosome segments down to approximately 10 megabases, enabling characterization of complex genomic changes. Lastly, fluorescence in situ hybridization, FISH, is a hybridization-based assay like microarray, but is almost always run on whole single cells. Fluorescently labeled probes bind to complementary sequences on sample nucleotide molecules, permitting strategic multiplex designs for targeting specific loci or entire chromosomes. Of the techniques discussed, FISH, specifically metaphase FISH, is most similar to DGH and can serve as a starting point. Metaphase FISH begins with live cells in culture. Not long before culture harvest, cells are exposed to a mitotic blocker such as coulsomid, which stops dividing cells from progressing past metaphase in the cell cycle. Cells are next dropped onto glass slides, where many of them will lyse, spilling their contents. Controlled ambient conditions cause those lysed cells to form metaphase spreads. Labeled probes are applied to the slide. If the sample nucleotides are DNA, heat and formamide may be used for denaturation. Probe DNA strands will then have access to anneal at their target sites and be detected via fluorescence. In this example FISH hybridization image, the probes target next to the telomeric repeat sequences of the chromosome 21q arm. Each of the 2 chromosome 21 homologs is composed of 2 chromatids, and each chromatid bears a fluorescent signal at the probe set's target locus. The basic FISH assay is the backbone of directional genomic hybridization, DGH, as can be seen in the similar workflow. As in FISH, the first step has cells in culture, but analog nucleotides are added to the culture medium before cell cycle arrest. The cells will incorporate the analog nucleotides as they synthesize new DNA strands in preparation for division. Those analog substituted daughter strands will be damaged upon later exposure to UV light and degraded by exonuclease, leaving each chromatid with only a parent strand. The probe applied to the sample targets either the negative or positive parent strand, but not both, as in FISH. These are the 3 principal steps at which DGH differs from FISH. Analog nucleotides are applied to the culture medium. substituted strands are exposed to UV and exonuclease to degrade them, and a probe targeting only one strand is applied to the sample. The rest of the steps resemble a typical fish assay. The larger figure on the left shows an example image of a DGH hybridization. It uses the same chromosome 21 subtelomere probe as before, which can be seen in the bottom right image. To summarize, a DGH hybridization requires both the sample and the probe set to be single-stranded. Sample DNA must lack daughter strands, and probe molecules must target only one of the parent strands, not both. To illustrate the signal pattern difference with DGH, we'll first look at the two pairs of chromosomes in the graphic. The pair on the left underwent a fish reaction, and the pair on the right underwent DGH. In each pair of chromosomes, the right-hand one has experienced a small inversion. In the fish reaction, this produces no change in signal pattern because both DNA strands are present and bound by probes. However, the single-stranded DGH example shows that while the green probe hasn't changed, the red signal is now on the opposite chromatid due to that inversion. In the side-by-side on the right, both chromosomes have an inversion in the P arm, but the green paint probe just covers everything in the fish reaction. Only DGH shows some of the fluorescence having followed its target DNA onto the left-hand chromatid. Making single-stranded DGH probes starts with bioinformatics. Traditionally, fish probe sets have been created using bacterial artificial chromosome BAC libraries, resulting in double-stranded probes. For DGH, bioinformatics sequence selection happens in silico. Candidate probe sequences with off-target complementarity or problematic binding characteristics are screened out from the start. Ideal sequences, all targeting one strand only, are then manufactured synthetically and fluorescently labeled. This production process means no repetitive DNA blocking agent, such as COT1, is required. For maximized signal-to-noise ratio, the maximized signal-to-noise ratio enables targeting of loci orders of magnitude smaller than with many BAC-based fish probes. Next, we can look at the three primary DGH tools: Screen, Discover, and Insight. All three generate single-cell data through direct visualization. Together, they enable the user to calibrate data from completely unbiased to highly targeted, depending on experimental needs. In Screen, the entire chromosome set is targeted at once. The single-cell nature of the images means variant data exists in the context of whatever else is happening in that specific cell. Abnormalities are confirmed through direct visualization. Screen can measure all rearrangement types detectable by FISH, array CGH, or G-band analysis, and also small inversions. Depending on local probe coverage, the lower limit of detection for Screen can be as low as 10 kilobases. This means even those rearrangements traditionally described as cryptic can be picked up. One exciting application of Screen paints is the role chromatid played as part of the NASA Twins study. Identical twin astronauts, Scott and Mark Kelly, were studied over the course of 1 year, while one was in orbit and the other was here on Earth's surface. NASA scientists needed a better way to track DNA damage suffered through exposure to the ionizing radiation of space. The DGH paints enabled the space agency to compare the frequency of structural rearrangements and other changes seen in the DNA of both subjects. Chromatid continues to work with NASA in collaboration with Dr. Susan Bailey at Colorado State University. This is the banding pattern you'd see with Discover if it were applied to the whole genome. It is generally applied in a more localized fashion. Within the region being targeted, this assay is unbiased, detecting all rearrangements present there. Breakpoints can usually be localized to within a single megabase, or even depending on proximity or even less depending on proximity to a band junction. The lower limit of detection is equal to that for screen. Discover is a powerful tool for projects seeking biomarker discovery or target validation, such as with cancer or rare diseases. DGH Insight assays generate locus-specific data, down to a resolution as low as 2 kilobases. In the image on the left, 3 high-value CAR-T engineering targets are measured at once. Keep in mind that while these may look like simple fish enumeration probes, each DGH Insight probe is an enumeration, break apart, and dual-color dual fusion probe simultaneously. The image on the right is a 3-color assay used to measure the outcome of a gene editing experiment. The assay simultaneously measures background genomic instability with the paints, insertions per cell with the yellow signals, location and orientation of those inserts, and insertion-related structural rearrangements. The green signals represent the target sites. A complex abnormal cell such as this one can represent a serious challenge to many assays. Array CGH generates gain and loss data, so it wouldn't detect balanced translocations. G-banding would be very hard-pressed to catch small insertions, as well as several of the small inversions present. Sequencing could be used to detect very small variants. However, data quality depends on the specific sequencing platform, read depth, and alignment algorithm, especially in highly repetitive regions. Without any bioinformatic intermediate step like sequence alignment, DGH enables direct visual confirmation of data. Now that we looked at a single very abnormal cell, let's turn to an example of the value of high-throughput DGH. Here is a polyclonal sample in which each clone has a different set of editing outcomes, but clones C and D have experienced tumor suppressor gene deletions. Clone D, being a very low prevalence population at 1% of the total, represents a serious risk to patients. The high cell count, yet single cell data generated by DGH enables direct detection of small populations, such as in this case. One of the benefits of sample sets is that variants detected may enable inferences about potential sources of abnormalities. The ability of DGH assays to generate data on genomic stability and insertional outcomes through direct visualization makes the technology ideal for this application. Reliable data derived from sample sets enables comparison of all aspects of an editing project. Donor samples may be compared against each other, the impact of various culture conditions, and the components of the editing method itself can be tested. The impact these variables may have on genomic stability can also be measured over different time points. Valuable inferences may be derived about potential sources of changes to chromosomal structure with direct single-cell visualization. Orthogonal data refers to information gathered through analysis of the same subject via unrelated methods. In this analysis sequence, samples are assayed first via G-banding, as it targets the whole genome of single cells in an unbiased way and is relatively inexpensive. Subsequent analysis by DGH screen, also single-cell, unbiased, whole genome, offers a higher resolution orthogonal comparison of the G-banding data. DGH Insight may then be performed on target loci identified by the unbiased assays. The high resolution of Insight can locate sequences as small as 2 kb and reveal structural changes like inversions within those sites. Lastly, sequencing can then be targeted to loci of concern, generating results which can be compared against existing data. Some techniques generate images which bear a striking resemblance to DGH assays. But let's look at how DGH data is completely different. Here are DGH screen on the left and spectral karyotyping, or SKY, on the right. If we look at screen first, we see 12 white arrows, each indicating an abnormality. These include aneuploidies, deletions, translocation, inversions, and sister chromatid exchanges. In the Sky karyogram, 8 out of 12 of those arrows, 2/3, are red, because Sky detects nothing abnormal there. They indicate data which has been left on the table by not using DGH. Which genes have been affected by those rearrangements? How will they impact patient safety and regulatory approval? Here's a screen image we saw earlier. If Sky alone were used, only the 3 rearrangements indicated by yellow arrows would be detected. Being based on FISH and labeling both strands at once, rearrangements that don't change chromosome size, number, or arm length will go unnoticed by Sky. While a FISH break-apart probe might be designed to catch individual inversions, that requires knowing in advance where the rearrangement will take place. Conversely, all the DGH assays are unbiased in the sense that they detect whatever is happening within the targeted area, be it a whole chromosome or a 2 kilobase insert. Established cytogenetics methods enjoy widespread use, but many other technologies, such as sequencing platforms, offer valuable data. They vary in cost, technical expertise required, how laborious or time-consuming each one is, their reproducibility, and excluded sample types. However, DGH technology offers a unique view of the genome. DGH data is based on single cells. The data output can be whole genome or locus-specific to the degree required, and results are based on direct visualization. Critically, the single-stranded targeting approach enables detection of rearrangement types not picked up by many other methods, especially at high resolution. These features make DGH assays an excellent option as a standalone suite or as a source of comparative confirmatory data alongside other platforms. Chromatid's senior science team collaborates with labs to craft the best analytical strategy, making the most of all available tools and data to accelerate scientific progress. At this point, Aaron Cross, Vice President of R&D, and myself would like to welcome any questions you may have. So can you explain a bit more about analog, the analog that is incorporated and how that works? So these are nucleotides that are added to the culture medium that are going to make the daughter strand more susceptible to UV irradiation later on in the processing step. It is added to specifically to increase that vulnerability and cause nicks in those daughter strands so that subsequent exposure to exonuclease can degrade them, leaving only the parent strands, so you can get the DGH outcome. Okay. At the recent FDA workshop on gene therapy products, the need for a deeper look at structural variants than G-banding was discussed. How does DGH fit in with some of the other methods out there, like optical genome mapping, bionano, and karyostat, for getting more comprehensive and resolved structural variant data? Want to take that one, Aaron?
Sure, I can handle that one. Great question. I would say that DGH fits in with these methods and can be used as the cytogenetic ground truth when these methods struggle with complex and heterogeneous structural variants, especially those with breakpoints in repetitive regions. Both methods mentioned are essentially extrapolating a picture of what the genome looks like in silico from measurements based on sequence alignment. Utilizing a whole genome DGH approach or a more targeted approach, as Ivan outlined, can be coupled with sequencing and array-based methods for variant calling and validation, especially for more complex variants, unstable variants, and highly heterogeneous variants. DGH screen is a great sub-in for G-banding, providing a much higher resolution around small inversion variants and complex variants, especially. Coupled with long-read sequencing methods and array-based methods, precise breakpoint identifications become possible. It can also be used to confirm or validate confounding or low-confidence sequencing results. As a package, these methods complement each other and provide a virtuous feedback loop, enabling researchers, I think, in probably the best way possible.
Thank you. So the next question deals with distinguishing between homozygous and heterozygous variants. So this kind of dovetails a little bit on the question that Erin just answered, which is that direct visualization. Chromosome abnormalities that are specific to individual chromosomes can be distinguished because it's direct visualization, because it's single cell. There's no mingling of DNA from different cells, and there's no averaging out of low prevalence variants or anything like that. Next question. I'm wondering how variable the efficiency of analog incorporation is among the different cell lines. Additional question, is your assay targeting only dividing cells? So that's going to be a yes on the dividing cells for DGH. And would you like to take that?
Sure. I mean, there's two aspects of that question. One is going to be just the timing of the cell population. If things are synchronized and you know the doubling time, it gets a lot easier. Most populations we look at with this method are asynchronous. They're a mixed population, and they have, you know, a pretty well-known doubling time, a 24-hour doubling time, an 18-hour doubling time. And in that way, the variation really lies in how long you expose the cells to analog. There are definitely cell lines that have special media and special media additives that can sometimes interfere with the uptake of the analogs that we use for DGH. So talking with our technical experts and making sure that our analog is compatible with your systems. We've only found one or two cases where we needed to change up the culturing conditions of the cells in order for effective uptake of the analog to occur. I would say that while it's a really good thing to talk upfront about your cell culture conditions and make sure that these two things are compatible, by and large, the method is easy. And as long as you know a little bit about your cell culture and how fast it's proliferating, the method is quite easy and most of the time goes off without a hitch.
And just to add to that, at this point, we've got pretty well-established quality control steps that take place at the bench. You know, when the analog incorporation is overshot or undershot a little bit, there are specific visual characteristics of the cells that are very easy to spot and we're very familiar with. And the culture setup protocol makes sure that you're aiming for your expected time, but you're going to catch the best time if it's a little bit over or a little bit under. So it's basically built into the protocols at this point. Thank you very much for all these great questions so far. Okay, what is the ballpark cost for the DGH screen assay? Does the potential user just need to send you frozen cells as a service? So that's definitely going to be something that the user has the option to send us cryo bullets where we will we can take the culture, rescue it, get those cells dividing again, and run the assay in-house as a service. We're also currently actively looking for potential beta testers that might be interested in performing the assay in-house themselves. As far as the ballpark cost, that's going to be dependent on number of cells, number of samples, what other factors?
It also depends on how many, what your cohort size is. So yes, number of samples, but we offer a couple of types of report, and this is dovetailing in with the second part of the question that you asked, which is what are the deliverables? You know, these assays can be used as to generate comparative data with a set of known controls, as Ivan discussed, and what is the difference in the genomes at defined loci and then all across the genomes from those defined controls. They can also just be used as an exploratory assay, which delivers a set of results around all types of structural variants. So it would be rates of inversions, rates of translocations, aneuploidy, both gain and loss, complex variants, any chromatid type aberrations, breaks, etc., dicentrics, and then also things like chromothripsis and micronuclei. All of those things will be given not only at a per chromosome level, but a per cell level. So anything where there's a scene of multiple variants in one cell, that the rates of variance in those cells will be comprehensively outlined in a report. We also offer karyogrammed images of DGH screen for each of the cells analyzed, if those are necessary for review by any individual researcher. Our assay format is defined, but our reporting format is quite flexible. So usually we wind up working with people on their reporting needs and what it is they're after, and deliver the data on the things that are most critical and most important for them to monitor. So basically, you know, it can be laid out in any way, shape, or form where we're teaching you how to do the cell culture yourself, and you're adding the analog and shipping us fixed cells. And we're running the assay and providing data, or you're shipping us your cells and we are doing the culturing and the prep and running the assay and providing you your data. And as Ivan alluded to, we're also looking for people who want to run the full thing themselves and even do the imaging, and we would partner with them for the analysis after that. So all things are possible. Please put an inquiry through the website if you'd like to learn more about pricing for very specific applications. We're more than happy to work with you to determine, you know, what the best type of assay and, you know, how that looks for your specific application.
And just to add to that, again, on the website, there are downloadable sample reports for all our different service offerings. To reiterate, Aaron mentioned that the reports can be customized based on the user's needs. So those downloadable sample reports are not sort of the end-all be-all. They're just an example, but it'll give you an idea of what those deliverables are. And as far as the beta testing opportunity, the one thing I would like to add also is that the DGH assay is pretty much just a fish assay. It's going to be using all the same hardware and reagents and everything. And the one different step is that UV exposure, but we can even work with folks if they have needs regarding the hardware for that. So what combination of these assays is best for assessing transgene insertions? So the assays chosen are definitely going to be catered to each individual customer's needs, to each set of project needs. the one aspect of DGH probes that was mentioned was that they are bioinformatically designed. They're synthetic oligos. So that's going to let us that's going to allow us to target very small loci, including transgenes. So we can put a probe on a transgene and literally just count the signals and locate wherever they're going. We can put a probe, and now this would be insight. We can label the target with a separate probe and assay background structural rearrangements that may be occurring to gauge genomic instability potentially related to the editing method using paints. So, and it really depends on which questions which questions need to be answered for the experimental needs of the project. Did you want to?
Yeah, just sort of to add on to what Ivan was saying, transgene tracking with our probes, we can get, pretty good coverage even for small targets, usually because transgene sequences are full of unique sequences. So as low as 2 kb, we've even gone smaller. And you can track these variants or these insertions across the genome. They can be occurring at a specific locus or expected at a specific locus, as Ivan mentioned, or randomly. And we're perfectly well equipped to track them in both ways. On and off target assessment is one of the things that we provide regularly for transgene insertion, as well as inverted transgene insertion against a specific locus or translocations alongside a transgene inversion at a specific locus. Those are some of the types of data. And this would all be DGH insight. We are currently working on developing an assay which couples DGH insight with a transgene and screen. That's something that's in development and something that people have shown interest in as well, in order to track purely off-target rearrangements alongside the impact of transgene insertion. So you're looking at the rest of the genome alongside the transgenic integrations with that combination of assays. And other than that, I think, we can always just do a count of transgenes that are inserted if it's a lentiviral system or AAV-directed system, and there are multiple inserts expected at variable copy numbers. We excel at giving not just an average copy number per cell, but the true count of how many cells have 2 copies, how many have 3, how many have 10, sort of data. So please just reach out if you have a specific question about this type of assay. We do this type of work quite frequently for researchers.
And one final point there is because the DGH probes are targeting one chromatid, all of those insertion sites, you're not only going to be able to determine that the insert is present there, but the signal pattern of the probe signal at that site is going to indicate whether structural rearrangements have taken place there as well. For instance, if there's been an inversion, if what orientation the probe has inserted in, etc. So next question here. If the assay is done in-house, including imaging, what is required for data analysis, special software, etc.? So this, to some extent, depends on the assay. No assay is specified here. So if you were dealing with insight, it comes down to how many colors you're dealing with. We have some DGH insight probes on the website, so that is an assay that folks can run in their own lab. However, if we're running the assay in-house, we are going to be taking care of all of that. The deliverable in that case is going to be a custom report whose parameters are determined at the outset by customer needs and by ongoing discussions.
And there's not really any special, there is special software that we use. You wouldn't need any special software if we were running the full suite for you. our software consists of, for a screen, the ability to generate a karyogram from a labeled spread. So that's that organized look at chromosomes aligned numerically that Ivan was showing off earlier in the presentation. We score from those with a defined set of scoring criteria. And for insight, we work with you to develop a scoring procedure around a defined set of scoring criteria. So those things are worked out as part of the service is the specification around which scoring criteria you need to be run on your particular samples with that particular assay. So if you're looking for translocations, if you're looking for a simple count, if you're looking for anything else that's defined and you want to ignore other things, we've set up very specific scoring campaigns for both insight and combinations of paints in sort of a mini screen format where it's 2 or 3 chromosome paints instead of the full course of whole genome chromosome paints. These things are also able to be run in fish. We didn't talk about that much in this presentation, but in some cases it doesn't make sense to use a DGH approach. These probes can be used with an initial phase DGH probe approach and then used in nuclei to count events where you're not looking at a DGH approach at all. You're looking at counting dots inside of a nucleus. It's a pretty flexible arrangement of tools that can be coupled with one another to serve the whole host of needs of any given project. And if you were to do it on your own, you would probably require certain imaging, at least to get to a metaphase spread. We are looking at developing platforms where metaphase spreads that are imaged, say with screen at our beta tester sites, are then, fed into a software on the cloud, and the karyogram, that organization of chromosomes, comes out as well as the data analysis. So that'll be future chromatid. But currently we offer the whole thing from start to finish, and have all the software in-house to deliver on those things as required.
And that was a good point. This presentation really didn't touch on the fish capabilities of some of these methods that we have, but because because we're starting with the bioinformatic design of synthetic probes, one of the interesting benefits of doing it that way is that you don't need that back library. You don't need to have bacterial cultures in the freezer. You simply download the reference genome for whatever species you're interested in, be it dog, boar, alpaca, C. elegans, whatever it may be, and we can design a custom probe designed for whatever strategic need, whatever scientific question needs to be answered. So if you want to break apart, if you want an amplification probe, if you need whatever is needed to answer the scientific question in whatever species is needed, that can all be done because these are being generated in silico.
Looks like the last question. Okay. I don't see any more questions coming through. It looks like we've gotten through the whole set. Thanks to the audience. That was a great set of questions.
Thank you so much for joining, everyone. We're going to have a short survey at the end of the session. It's about 3 questions. If you have time, please fill it out, and we will let you know about our next webinar. Any of the assets that were mentioned today are at chromatid.com, so please visit us and send us an inquiry if you're interested. Thank you so much, everyone. Have a great day.